My group works on sensory and motor neuron diseases in both preclinical and clinical aspects. And I'm excited to tell you today about precision gene therapy for treating severe pain. As people know, chronic pain is a large and severe problem. It affects a full quarter of adults in the US, and 40% of those cases are high impact, meaning that there's substantial impairment in function and quality of life. The lack of effective treatment for pain fuels the opioid epidemic. And what people may not realize as clearly is that most severe pain conditions are focal, but almost all treatments are systemic and thus expose patients to unwanted side effects. Pain medicines in general have poor therapeutic indices, and this is particularly problematic for elderly patients and those with medical comorbidities. And actually, the funding supporting this work has come from the National Institute of Aging because the undertreatment of elderly patients is such a large problem. I'll just remind people that sodium and potassium channels play opposing roles in membrane depolarization and hyperpolarization necessary to fire action potentials. So the upward slope of an action potential is shaped by sodium influx through voltage-gated sodium channels, and potassium channel efflux terminates the action potential by hyperpolarizing the membrane potential. There's very strong human genetic data that argues that silencing primary pain-sensing neurons is sufficient to eliminate pain. So the NaV1.7 voltage-gated sodium channel is selectively expressed in primary pain-sensing neurons. There are homozygous NaV1.7 knockout humans who have congenital insensitivity to pain. So these kids experience no pain despite otherwise intact peripheral nervous systems. In contrast, there are heterozygous NaV1.7 gain-of-function patients, and these have severe painful conditions such as familial erythromelalgia, which is characterized by paroxysms of extreme pain in the extremities and redness and swelling. There have been major efforts by pharma and biotech to block or silence NaV1.7, but these have met with limited success due to the need for complete or near complete block or knockdown of the channel, and potentially also the need to access proximal nociceptor terminals in the central nervous system. A novel strategy is to use AAVs to deliver potassium ion channels using a spatially precise gene therapy method. There's human genetic data to support this strategy, so there are naturally occurring gain-of-function potassium channels that are disease modifying for familial erythromelalgia and other pain conditions. So this is a figure from a paper by Steve Waxman, and you can see that in black, there's the time in pain for typical patients with one of these rare pain conditions. On the right, in red, you see the reduction in pain due to a gain-of-function mutation in a potassium channel that's compensatory. So the strategy that we're proposing is to inject AAVs expressing potassium channels into target nerves. And so we really want a focally precise treatment because the potassium channel expression is aggressive. Inhibitory studies have shown that only 15% of sensory neurons need to be silenced for a large effect on pain. And we would expect a good safety profile because we're able to use low doses of virus, native expression promoters, and native proteins, as opposed to, in the case of AAV toxicity, high viral doses, strong viral promoters, and foreign payloads. So we take advantage of mice that express the light-sensitive ion channel rhodopsin, specifically in nociceptors, and blue light applied to the paw of these mice is equivalent to an injection of hot chili pepper, so it's very painful. And we inject the sciatic nerve of these mice with the potassium channel virus, and then several weeks later we can take out the sensory neurons and assay them in vitro, using a high-content calcium imaging platform that we've developed. And you can see that there's a marked reduction in the excitability of nociceptors following injection of the potassium channel gene therapy. So how might we apply this? The initial indication that we're pursuing is painful neuroma. So neuromas are benign growths of nerves, supporting tissue, and inflammatory cells following nerve injury or amputation. a majority of these are very painful, and painful neuroma affects over 300,000 people in the US with profound impairment in quality of life. There are very few medical or surgical treatment options, and we have a collaboration with Dr. Kyle Eberlin, who's a peripheral nerve surgeon at MGH and specialist in neuroma. And so the initial first-in-human studies will involve injection of the nerves that feed these neuromas and reduction in pain. And we would anticipate, though, that the clinical use of this application could be using ultrasound guidance, as is commonly performed for regional anesthesia. So we've shown benefit of this approach in several animal models, and these include a painful neuroma model in which several weeks after the virus injection, there's a strong reduction in pain, as well as an inflammatory model using complete Freud's adjuvant. And you can see on the right, there's almost an equivalent thermal pain sensitivity in animals treated with the potassium channels, but on the left, animals treated with control have a marked increase in pain. We believe that there's several additional opportunities for this application. So the technology can be expanded to other focal severe pain conditions. We have strong preclinical data for trigeminal neuralgia and believe this can be applied to focal compression neuralgias, shingles, and complex regional pain syndrome. And in order to use a broader delivery, we require additional precision. So it would be wonderful to be able to treat people with refractory low back pain through epidural delivery, but this requires additional precision. And we've done this through the development of cell type precision. So we've achieved this using short promoter sequences that restrict the potassium channel expression only to pain-sensing neurons. We identified genes that were nociceptor specific and actually increased in pain models, and then identified optimal segments of those promoters that fit alongside a potassium channel payload within the limited AAV cargo capacity. And this approach avoids expression in other sensory neurons and motor neurons. So we believe that through this dual cell type and spatial precision, we can use broader delivery strategies, and our animal data shows that we obtain a similar benefit in pain reduction. We also believe that the promoters enable a platform approach for the targeted delivery of other cargo to specific neuronal subtypes. So we believe that the AAV-based delivery of potassium channels can revolutionize pain treatment and transform it from using systemic drugs that have poor treatment indices to a dual spatial and cell type precision strategy. The initial indication of painful neuroma is very well suited, and that's because it's a well-defined population with a severe clinical need, few options, and an optimal anatomy for applying the technology. The technology can be expanded to other focal pain conditions, and then through the use of novel delivery methods and the customized promoters to platform applications for the delivery of other payloads. And for us, the next steps are lead optimization, in which we want to perform dose response and safety studies in rodents and non-human primates, and focus on biomarker development, including translational biomarkers of nociceptor physiology. And my group has performed and is currently performing studies of sensory nerve excitability in patients, and those technologies can be used in rodents, non-human primates, and people as translational biomarkers for this technology. We'll also want to begin process development for a generation of a high-quality virus. We'll want to begin formal IND-enabling studies in the end of 2024, and we're seeking partnerships to accelerate the development and commercialization of this technology. So I'd like to thank the people in my group who have performed this work, our funding sources, and everyone here for their attention. Thank you so much.